Hello, I am Shekinah Challam, Assistant Professor in the Department of Biotechnology, St. Edmund's College, Shillong. Today we're going to talk about sickle cell anemia. Sickle cell anemia is a life-threatening disorder, and it is a disorder that is passed on or inherited from parents to offsprings. So we also have people with sickle cell anemia have this blood disorder, and as a matter of fact, sickle cell anemia is one of the most common blood disorders that is known. Sickle cell anemia is found mainly in the sub-Saharan areas, and also India ranks second in that list. But with migration of population, we also see that the sickle cell gene is spread all over across the globe. In India, we have Chhattisgarh with the highest number of sickle cell cases, and Chhattisgarh also has an institute, a sickle cell institute, to study very deeply the causes and the precautions, cures and treatment for sickle cell. Besides Chhattisgarh, it's also found in Orissa. High cases are found in Orissa, in Madhya Pradesh, Maharashtra, Gujarat, Andhra Pradesh, Telangana. A few cases are also found in Tamil Nadu and Kerala. Now, sickle cell disease is actually a disease that involves the function of the red blood cells. Now, the red blood cells are present in the blood, and one of the main functions of the red blood cells is to carry oxygen to the various tissues and carry carbon dioxide back to the lungs. And the problem with sickle cell anemia is that these RBCs, the structure of these RBCs are compromised. We will find that RBCs are normally biconcave in their shapes, and they are also packed with a lot of hemoglobin. But in sickle cell anemia, this hemoglobin has become abnormal, and therefore will force the biconcave discs or the RBCs to change their shape into the sickle shape or the crescent moon shape. So we will look and find out about the structure of the hemoglobin. Also, sickle cells do not last in the body as long as normal RBCs do. Normal RBCs survive for about 120 days, whereas sickle cells survive only about 10 to 20 days and are getting destroyed, as a result of which it leads to a condition of anemia, hence the name sickle cell anemia. Now, a normal hemoglobin looks like this, having two alpha chains and two beta chains, and an abnormality in sickle cell anemia is present in the beta globin chains. Now, the beta globin chain, the genes for the beta globin chain is present on chromosome number 11, and there is a mutation that has taken place in the 6th position of the 146 amino acids that are present in the beta globin chain. As you would see in the diagram, there is a mutation in the 6th position, and if you look at the sequence over here, GAG is a normal DNA sequence. It's just a partial sequence, and which would normally code for glutamine. This is the 6th position of the beta globin chain. But because of this missense or a point mutation, we will now find that GAG is now replaced with GTG, and that codes for valine. So GAG present in a normal DNA sequence has mutated to GTG, therefore replacing glutamine with valine. A normal hemoglobin molecule looks like this, whereas because of this mutation, the hemoglobin molecules now begin to polymerize, as a result of which they distort the shape of the red blood cells. Here is a diagram that will show you what abnormal hemoglobin looks like within the red blood cells. So because of the polymerization of the abnormal hemoglobin, it will force the shape of the red blood cells to take up this shape, that is the shape of a sickle. As a result of which, you will find that these sickle cells are not very flexible, and they will begin to block the flow of blood in smaller blood vessels such as the capillaries. So because of this, there will be various clinical manifestations of the disease. There will be a pain crisis where the individual suffering from sickle cell anemia will experience a lot of pain. There's of course anemia because the red blood cells do not last in the bloodstream as long as they should. They last only about 10 to 20 days in contrast to the normal RBCs, which survive for about 120 days. There's also the problem of jaundice because these sickle cells are getting destroyed, and the bilirubin that's accumulating because of their destruction is not being removed by the liver as quickly as it should for the normal RBCs. Therefore, the individuals suffering from sickle cell anemia tend to have a yellowish tinge in their eyes, in their lips, and in their skin color. There's also dactylitis, which is a pain of the joints, swelling of the joints, the hands and the feet. Persons also suffering from sickle cell anemia are very prone to infections. There's delayed growth and development, especially in children. They may have vision problems, and in very rare cases, the individual may also go blind. You also have organ damage because of what I have shown you earlier. The sickle cells begin to clog up and block the blood vessels, resulting in kidney damage and also damage to the heart. We also have a condition called splenic sequestration, where a lot of these RBCs accumulate in the spleen, and besides that, individuals with this disease also do experience a shorter lifespan, unfortunately. This disease may be detected at birth if a screening is done, but if no screening is done at birth, perhaps when the child is about the baby is about 6 months old, when the fetal hemoglobin has been washed out, and then you have the adult hemoglobin HbA that is present in the blood. That is perhaps the time when it will be detected in an individual. There are others. The tests may be performed by a simple blood test, and there are various techniques for doing it. Modern techniques involve DNA sequencing, RFLPs and PCRs, but a normal blood test will also be able to tell the disease, to tell if the patient has the disease by simply looking at the shape of the red blood cells in that individual. Now, like I said earlier, sickle cell anemia is an autosomal recessive disorder, which means that individuals who carry the sickle cell allele will have to have that allele in the homozygous form. So I will take you to a Punnett square to explain what this looks like. So if we are going to represent a normal individual with A and A, that is hemoglobin A, normal hemoglobin A represented as A, and the sickle cell allele is present as S. So this individual that we're looking at over here has normal hemoglobin A. So both alleles present are normal alleles, and if such an individual should cross with another individual who has the sickle cell gene. Now this individual having the sickle cell gene is represented as A and S. That means this individual has the sickle cell gene, but in the heterozygous condition. Such an individual will not present the disease, but will be a carrier of such a disease. So this individual will have normal hemoglobin in one of the alleles would be normal, whereas the other one would be sickle cell. If such a cross is made between two such individuals, the offspring will have one of the four possible combinations. 50% of them will be absolutely normal, whereas the other 50 will be carriers of this disease. They will present no physical manifestations of the disease. As a matter of fact, these individuals have been found to be resistant to malaria. It has been thought that this sickle cell anemia may be an adaptation by the human race to resist infection by the malarial parasite Plasmodium. So these individuals will have sickle cells, but will be carrying the sickle cell gene, but will not be manifesting the disease. Now, in the second situation, if a normal individual is made to cross with an individual who is diseased or who is presenting the disease, such an individual will have S and S. These are present in the homozygous condition. A cross such as this will result in this in offspring who are actually just carriers of the disease. They will not present the disease, but they will be carriers. Now, if these carriers were to mate, so a carrier is mating with another carrier, and these are the possible outcomes. One of the offspring will definitely be having the disease or will have the sickle cell condition, whereas the 50% will be carriers of the disease and 25% of the offspring will be absolutely normal individuals. In the fourth situation, where we have a carrier breeding or crossing with a sickle cell disease, a person having sickle cell disease, 50% of the offspring will be present, will be carriers of the disease, where the disease will be present in the other 50. They are represented as SS and SS, homozygous condition. In the last possibility, if a carrier represented as AS were to cross with an individual having the abnormal hemoglobin C, unable to properly carry oxygen to the various tissues, in such an individual, if the allele C is present, 25% of these offspring will express the disease, will suffer from sickle cell disease, and 25% will be absolutely normal, and the other 50% will be 25% will be a carrier of the disease, whereas this other person will also not manifest any symptoms of the disease. So they used to say we Indians have marriage kunlis. It has been suggested that perhaps people can also think about having marriage kunlis using sickle cell disease traits to make sure that the children do not suffer from sickle cell disease or sickle cell anemia, which is a life-threatening condition. Now, what are the possible treatments for sickle cell disease? First one is blood transfusion, to make sure that the individual will be able to have the right amount of hemoglobin in their blood. People with sickle cell disease, like I said earlier, are very prone to infections. Therefore, treatment with penicillin is suggested. Hydroxyurea has been used over the years to prevent any complications like the one we described earlier. A almost sure shot treatment for the disease is a bone marrow transplant, which is of course quite difficult to get. But should an individual get a match for a bone marrow transplant, the individual should go forward with this, because that is a sure treatment for the disease, to make sure that the person does not present any more symptoms of a sickle cell disease. Gene therapy, one of the most promising fields and advancing fields in biotechnology, is perhaps the answer, a sure shot treatment for sickle cell disease. But gene therapy is in its developing stages and also very expensive. So in gene therapy, what they do is really whatever mutation that we have talked about earlier, in that mutation will be corrected, and all the cells that arise from these treated cells, gene therapy treated cells, will no longer carry the sickle cell gene, and the individual will be absolutely healthy. We are hoping that the gene treatment for sickle cell disease will be available to all individuals suffering from sickle cell disease very soon. But because there is no, like I said, no concrete evidence to point that we are getting treatment or cure for sickle cell disease, individuals will still have to manage the disease on a day-to-day basis. The first one obviously would be medical compliance and regular medical care as advised by the physician. Avoiding extreme temperatures, because extreme temperatures tend to aggravate the sickle cell symptoms. Infection prevention by taking vaccines. Staying hydrated is very important. Eating the right kind of food, making sure that your food is rich in iron, that is also very important. Physical activity will ensure that the blood is very well circulated in the body of the individual suffering from sickle cell disease. And of course, it is very important to have an emergency plan should things get complicated and complications arise. There should be an emergency plan to contact the treating physician and to get the help that is needed. So up until now, I think individuals with sickle cell disease must also manage the disease well. Before I forget, I think individuals with sickle cell disease must avoid very high altitudes, because we know the higher the altitude, the lesser the oxygen that is present, and as it is, they're not very capable of trapping that oxygen in their bodies. So that is it for now. Thank you.